Today we see about mitosis and meiosis. Mitosis. Mitosis is the process of cell division wherein the chromosome replicate and get equally distributed into two daughter cells. The chromosome in the number is equal to their parent cell, that is diploid. Hence the mitosis known as equational division. Mitosis consists of four basic phases: prophase, metaphase, anaphase, and telophase. Breaking prophase into early phases called prophase and late phases called prometaphase. These phases occur in the strict sequential order and cytokinesis. The process of dividing cell contents to make two new cells starts in anaphase or telophase. Interphase, the mostly active growth of metabolism in the cell, but the end of interphase DNA duplicates. During the interphase, chromosomes are referred to as a chromatin, a loosely scattered arrangement of chromosomes. Prophase. At the end of the interphase, the chromosomes replicate themselves. This signals the start of prophase. During the prophase, the centrioles migrate to the opposite folds of the cells. The nuclear membrane breaks down and dissolves. The chromosomes are shortened and thickened. At this point, replace their parent chromosomes are called chromatids. The chromatids are joined together in a region known as the centromere. As the prophase continues, some of the cells are cytoskeleton, form a spindle fibers. By the end of prophase, the entire network of microfilaments has formed and it is attached to the chromatids and the centromere. The cell now enters to the metaphase. Metaphase begins as the chromatids begin to move towards the equator of the cell when all the chromatids have reached the center of the cell and begin to separate. Next, the anaphase begins. Anaphase begins with the separation of chromatid pairs. Once separated, each chromatid is called chromosomes. By late anaphase, an equal number of chromosomes has reached each pole. The cell membrane begins to change the shape, getting and thinner in the middle. Telophase. During the early telophase, the chromosomes are uncoiled. The fragments of the nuclear membrane begin to resemble around the chromosomes. The nucleolus reappears. The cell membrane and the cell plate in plant cells begin to form in the space between the two nuclei. Next, meiosis. Sexual reproduction in organism takes place through the process of fusion of male and female gametes that are produced in the form of sperm and egg respectively. Gametes are normally haploid in nature, which means that they only contain about half the number of chromosomes. This genetic content tends to make different form of all other types of body cells that are present. It is the process of meiosis that produces these haploid cells. Phases of meiosis. In each round of division, cell experiences of four phases of meiosis 1 and 2: prophase, metaphase, anaphase, and telophase. In meiotic, prophase 1 is subdivided into 5 stages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Prophase 1. Prophase 1 is the longest stage of meiosis. During the phase of the prophase 1, homologous chromosomes pair and exchange DNA. This frequently results in a chromosomal hybrid. The combination of DNA made during the hybrids is a critical source of heredity and variation and results in a new mixes of alleles, which might be beneficial. And the crossing over is at the chiasmata. Leptotene, the first stage of prophase 1, where the chromosomes are condensed and become visible as the thread-like structure. Next, zygotene. Homologous chromosomes from the mother and father are come together and form a pair. Next, pachytene. In this stage, the recombination or crossing over takes place. Next, diplotene. The synaptonomal complex are vanished towards the conclusion of diplotene synapses, although the homologous pairs of remain linked at the chiasmata. Next, diakinesis. Chromosomes become fully condensed and the nuclear and membrane and nucleolus are break down to form the next phase of meiosis 1. Next, metaphase 1. The cells senses this strain and does not progress with the anaphase until the chromosomes are appropriately bisituated in meiosis. The homologous chromosomes are aligned on either side of the equatorial plate. Next, the anaphase 1. Kinetochore microtubules become short, pulling homologous chromosomes to opposite poles. Non-kinetochore microtubules extend, pushing the centromeres separated apart. The cell stretches for division down the center. Next, the telophase 1. The primary meiotic divisions adequately close when the chromosome touch base at the poles. Each daughter cell presently has half number of the chromosomes. However, every chromosome comprises the pair of chromatids. The microtubules that make up the spindle network break down the another nuclear membrane encompasses very haploid pair. The chromosomes uncoil once again into the chromatin. Cytokinesis, the pinching the cell wall of the animal cells or the development of the cell wall in the plant cells happens, completing the formation of two daughter cells. Sister chromatids stay connected during the telophase 1. Meiosis 2. Meiosis 2 is a mitotic division of each of the haploid cells produced in the meiosis 1. Prophase 2. The nuclear membrane and nuclei break up while the spindle network appears. Chromosomes do not applicate any further in this phase of meiosis. The chromosomes begin migrating to the metaphase 2 plate. At the end of prophase 2 of meiosis, the cell enters into a metaphase 2. The metaphase 2. In metaphase 2 of meiosis, the chromosomes line up at the metaphase 2 plate, the cell center. The kinetochore fibers of the sister chromatids pointed towards opposite poles. At the end of metaphase 2 of meiosis, the cell enters into the anaphase 2. Next, the anaphase 2. The sister chromatids separate and begin moving to the opposite ends of the cell. Spindle fibers are not connected to the chromatids length of the elongated cells. Once the paired sister chromatids separated from one another, each is considered as a full chromosomes. They are referred as a daughter chromosomes. In the preparation for the next stage of meiosis, the two cells of the poles also moves further apart during the course of anaphase 2. At the end of anaphase 2, each pole contains a complete completion of chromosomes. Following the anaphase 2 of meiosis, the cell enters into the telophase 2. Next, the telophase 2. Distant nuclei form at the opposite poles. Cytokinesis, division of cytoplasm and the formation of two distinct cells occurs. At the end of meiosis 2, 4 daughter cells are produced. Each cell has one of the number of chromosomes as the original parent cell.